Hey folks, this video will look into the topic of conservation biology, which is a deep topic and somewhat troubling inherently. And we could easily spend an entire semester only focused on conservation biology. So this video will serve as an introduction to this very important branch of biological sciences. And before we really get into understanding what conservation is all about, I think it's important to take a moment to think about the definitions of some words that come up often in the context of conservation thinking. And these are words like nature or natural, of course, the word conservation itself, and the word preservation. So what do these words mean? Well, you've probably heard all of them before, but I don't know how long it's been since you sat down and looked up the definition of these words, if ever. So nature is a word that gets thrown around all the time. And if we were to look at a dictionary, what we would find is the definition of the word nature is the external world in its entirety. Okay, whatever that means. Or essence. Okay, that's even more vague. Or we could talk about the word natural, which is rooted in the word nature. And so what are we talking about when we're talking about nature or things that are natural? A lot of times folks would say when we're talking about things that are natural, we're talking about things that are happening in the absence of any human influence. And for the most part, that definition seems to work pretty well. But it's also important to recognize that we ourselves as a species are a naturally evolved lineage, and we are a natural component of the universe. Natural. That is, we are an inherent property of the universe also. But frequently, when we're talking about nature, we're talking about really the entire external world in its entirety beyond just simply human perspective. So we could talk about the entire universe and how long the universe has existed and all the things that have happened during the existence of the universe. And there's a lot going on behind the scenes there, but we can apply that thinking to biological systems. How long have biological systems been around on our planet? What kinds of natural or what kinds of processes have biological systems gone through that occur in the absence of any human intervention, or which kinds of those processes influence human existence? And so, obviously, things that happen in natural ecosystems or undisturbed ecosystems, those that are undisturbed by human activities anyway, are very representative of what sort of happens to biological systems in the absence of human influence. So that's one way we could think about this word nature. The word essence really does refer to the individual, real, or ultimate nature of a thing. It's a little bit confusing to have the word essence in the definition of the word nature when the definition of the word essence includes the word nature. That's sort of this circular logic. But nevertheless, if we think about essences, which kind of goes back to thinking about like ancient Greek philosophy, essences really do represent what things truly inherently are, all of their inherent properties, their real ultimate characteristics are their essences. The concept of conservation is again rooted in this word conserve, and to conserve is to avoid wasteful or destructive use of something. And that's a little bit more of an intuitive definition. We could think about conserving water or conserving natural resources in the environment, say, again, in the context of how we use them as humans, as a species. But if we are conserving water, we are avoiding wasteful use of water. We're only really using it for necessary things, for example, or if we're conserving natural resources such as fossil fuels, we are trying to avoid wasteful use of those resources. And we're certainly trying to avoid destroying those resources. And so the definition of the word conserve gets translated into the definition of the word conservation, which is a careful preservation or protection of something. And so again, if we're trying to conserve resources, we are trying to preserve them and protect them to ensure that they continue to be available into the future. This gets us at, well, what's the definition of the word preservation? Well, preservation is the activity or process of keeping something valued alive, intact, or free from damage or decay. And so this really points at the important difference between the word conservation and the word preservation. Conservation is really doing things that make sure that resources are available into the future, whereas preservation is really the process of keeping something intact or keeping something from sort of becoming damaged or decaying or eroding away. So they are very similar words, but they don't exactly mean the same thing. So sometimes you may have been out in certain places that were called nature preserves. Well, what's a nature preserve? Well, if we think about the word nature and we think about the word preserve, we can put those two definitions together and get at an understanding of what a nature preserve is supposed to do. It is supposed to basically take an area that has a naturally occurring ecosystem or an ecosystem that exists independent of significant human influence and make sure that that ecosystem is allowed to remain intact in terms of all of the normal biological processes that happen within it for subsequent future generations to come. That would be the overarching goal of a nature preserve. We never see things called nature conserves or conservations. That's not really terminology that gets applied. Although obviously the overarching goal of having a nature preserve often is framed in the context of conservation. But nevertheless, there's a little bit of a difference between these words. So anyway, I think before we branch off into really understanding what the science of conservation biology is all about, it's pretty important to think about what some of these words mean because these words will get used a lot within this branch of biology. Now, conservation science really is an interdisciplinary subject. And so there are all kinds of branches of the natural sciences that are involved in conservation biology. And there are sort of non-scientific fields that sort of intersect with the field of conservation biology inherently. There are politics involved. There are certain aspects of natural resource management that get involved, which again, may or may not be so scientific. And when we sort of put together all of the different fields of thinking that are required in order for conservation biology to be realized, what we end up with is an understanding that conservation biology really is a crisis discipline. And that might sound daunting, but it really is. And often, why are folks involved in the conservation movement? Well, frequently the goals of conservation are preventing the utter loss of biological resources. And that often is framed in the context of some type of a crisis, like a species going extinct, or a whole bunch of ecosystems where different species might have naturally occurring habitat becoming destroyed or displaced. And these are sort of situations that we consider to be crises because there aren't really easy solutions to solving those kinds of problems. And there are tons and tons of goals in conservation biology. Again, a lot of them are scientific inherently, but in order to achieve those scientific goals, there does have to be some intersection with understanding how the overarching goals of human society can effectively really manage biological resources. And this leads to, again, this becoming this really interdisciplinary field. And oftentimes what ends up happening is that the goals of conservation biology might really be up here. Maybe we're shooting for some pretty high goals, but typically, and this is a sad reality, in the end, what ultimately gets realized often in conservation work is that what can be achieved with regards to those goals is really more realistically here compared to what the actual goal is or was. And so conservation biologists often have to deal with the limitations of what is actually practically achievable in order to conserve species or in order to conserve habitats for those kinds of different species that might be threatened, for example, by human activities. And a lot of that's because not all stakeholders involved in managing natural resources necessarily all agree about how those resources should be managed. And a lot of that has to do with politics and economy, because natural resources often are worth money. And exploiting those resources for their monetary values often comes at a cost for conserving biological resources. And so biologists and business owners or politicians or legal type people often have to meet at some kind of a middle ground where both parties or all parties involved are sort of equally appeased with what the outcome of a particular management strategy might be. And again, so if the ultimate goal of conservation is here, probably what we're going to realize when we take action is something more here. And when the ultimate goal, likewise, of some political organization or natural resource management agency is here, what they might ultimately see realized might be down here. And this is happening for both parties involved because they both have to have their desires and their goals appealed or appeased, and that often involves some level of compromise. So there are lots of different combined efforts related to conserving and preserving natural biodiversity that again exist across a whole spectrum of different fields, from those that are very, very scientific to those that are a lot more political. Now, when we do talk about conservation biology, we are often concerned with a pretty important central theme, and that theme is conserving biodiversity. Now, this is easily said, but understanding what it really means is a bit more nuanced. What truly is biodiversity when we talk about this term? Well, biodiversity can be measured in different ways, and it can be measured at all different levels of biological organization. So if you recall, the most fundamental level of biological organization is the cell, and from the cell to the tissue, and from the tissue to the organ, from the organ to the organism, from the organism to the population of organisms, from populations to communities, from communities to ecosystems to the biosphere. So there are all these different levels of biodiversity or biological organization, and we can sort of quantify biodiversity at some of these different levels. And most broadly speaking, we can define biodiversity really as the whole sum of population genetic, species-wide, and community ecosystem diversity. What does all that mean? Well, at the sort of lowest level, when we're talking about population genetic diversity, we're really asking a central question: how much genetic diversity is there in a population of a species, and then among all of the different populations that make up that species, how much genetic diversity is there? So really looking at this at the level of genes kind of represents the adaptability of different populations amongst different species. When there's a lot of genetic diversity, that tends to correlate to a lot of adaptability. When we don't see lots of genetic diversity, that tends to lead to a lack of adaptability. And this turns out to be important for conservation, because if species are sort of not very genetically diverse, then they can't really adapt to change. And if they can't adapt to change very easily, then it becomes kind of hard to conserve them, particularly in the context of human resource exploitation. Species-wide biodiversity is a little bit more intuitive, I think, and that could be framed in the context of simply asking, how many species are there? And then we could ask, well, how many species are there in a particular community? Or how many species are there in different kinds of ecosystems? Or how many species are there in a particular geographic area? Or how many species might make up a particular kind of biome? And all these ideas kind of point towards something we call species richness, which is simply just a count of the number of species that occur in different areas. And of course, we could talk about global species richness. So how many species are there in the world? And I invite you to think about that question and perhaps even go and try to look up the answer. And I think what you're going to find, you try to look up the answer to the question, is that nobody really knows exactly how many species exist. And that's kind of odd, but it actually makes a lot of sense, because A, there's a lot of variation in the way that species get defined, and that depends upon these things we call species concepts. And B, there are populations that exist that have not been described yet, and that could be because the areas where they occur haven't been explored, or that could be because there is a lack of experts that study different taxonomic groups. And so the whole biodiversity of a particular taxonomic group might not be super well understood, because not a lot of people focus on those groups. Some groups might get a lot more attention than others. We might call groups that get a lot of attention in the context of conservation charismatic species, whereas groups that don't get a whole lot of attention in the context of conservation, not so charismatic. And so typically, large mammals are often viewed as charismatic species in conservation, whereas maybe things like plants or microorganisms that are important in the context of conservation don't get nearly as much attention. Anyway, species richness is really another measure of biodiversity above just the level of genetic diversity. And then we could talk about ecosystem or community ecosystem diversity. And that's really asking a question about to what extent is there variation in the kinds of communities or kinds of ecosystems that exist really across a geographic area or across the entire globe. And that turns out to be important because without a tremendous amount of ecosystem diversity, it tends to be hard for us to realize or to have a whole lot of species level diversity because different kinds of ecosystems are required for different kinds of species to have suitable habitat. And so all these things are very sort of intertwined in terms of their importance and their interdependence upon one another. Species level diversity really does depend upon genetic diversity, and ecosystem diversity kind of depends upon species diversity. And so it's kind of hard to frame the importance of one of these things without interpreting the context of the others. Now, again, when we start thinking about species-wide diversity, or the number of species that exist, as it turns out, the tree of life is not really shared proportionally by all the major taxonomic groups. Again, we don't really know how many species there are. We have some estimates. There might be something like maybe 500,000 different species that have been properly described, but that's still an estimate. And there might be situations where there are species that haven't been described, or where there are two different species that have been described, but in some cases, really those two species could be better described as a single species because they're not really reproductively isolated, for example. But I think the estimate of about 500,000 species or so is roughly accurate. And then we can look at how the biodiversity amongst these major taxonomic groups is partitioned. And what we can see is that about 59% of all described species are insects, which is kind of interesting. How adaptable insects have been throughout the course of their evolutionary history really speaks of how they have been able to diversify more than any other groups that we see by far. And among those insects, 37% of the described species are beetles that belong to the order Coleoptera. And so beetles are really the most, represent the most extraordinary patterns of diversification of any taxonomic group that we've seen on this planet. And they make up a really substantially large chunk of this sort of total number of described species on our planet. We can also look at the distribution of photosynthetic life. And so not surprisingly, knowing that photosynthetic organisms form the basis of all terrestrial and aquatic ecosystems, that kind of translates into how many of the described species are photosynthetic forms of life. And it turns out that about 18% of all described species are either plants or algae. And we can see them distributed on this little chunk of our pie chart here. And then we can see less so other organisms, such as the other invertebrates that are not insects, making up a pretty big chunk of the tree, about 12%. And then everything else, like vertebrates and fungi or microbes such as protists and archaeans and bacteria, really only make up a pretty small chunk of our tree. We see that fungi makes up about 4% of total biodiversity. Other organisms, these are other microbes, again, archaeans, bacteria, protists, make up about 6% of this tree. And vertebrates, which again are some of the most charismatic taxa on the planet, especially in the context of conservation, only make up about 1% of the entire number of described species. So this is pretty interesting, and we could ask questions about how different environments across the globe have really driven different patterns in the taxonomic distribution of biodiversity. And obviously, some parts of the globe have a lot more biodiversity than others. Particularly, we can think of the tropical regions as being biodiverse, but not exclusively. California, for example, is a place that also harbors tremendous amounts of biodiversity, particularly when we look at the diversity of plants in the state of California. And typically, as one starts to move towards the polar regions, we don't see nearly as much biodiversity. And part of that might have to do with the fact that productivity, ecological productivity, tends to decrease with distance from the equator. As I mentioned, species richness is a pretty standard measure of species-wide biodiversity, but it's a little bit of an oversimplified measure because species richness only accounts for the number of species, counts of the number of species, but species richness itself doesn't account for the abundance of species. And so if we account for both the richness of species and the abundance of species, we can get at a more accurate measure of diversity that represents not just how many species there are, but what is the relative abundance of different species in different areas. And that gets us at a better measure of diversity. And one such measure is H prime, known as the Shannon-Weaver Diversity Index. And the math isn't terribly crazy. It looks kind of crazy, but it's basically a brief statistical expression or statistical model used to evaluate biodiversity. And it's effectively the sum of the relative abundance of all of the different species that occur in a particular area, multiplied by the natural log of that relative abundance. And then because this number turns out to be negative, we end up making this number negative with a negative sign at the end. But effectively, when we add up the relative abundance times the natural log of the relative abundance of all the different species that occur in a particular area and multiply it by -1, we get a measure of diversity. So that's one way of evaluating diversity. It's not super useful if we only look at the diversity of one site. This metric is really designed to be used as for making comparisons of different areas, so we can compare the diversity of different sites to one another to get a sense of which areas have a lot of diversity, which ones tend to not be really dominated too much by a single species, compared to areas that don't have a lot of diversity that maybe are really kind of dominated by just one or two species. That's typically a measure of diversity taken at a single area. We can also talk about measuring landscape scale diversity. And so that's measured in the context of what we call alpha, beta, and gamma diversity. And this does rely simply on species richness, so counts of species. We could look at the alpha diversity of a particular site. That's simply how many species does a site have. This one here in this cartoon has 4, and this one here has 4, and this one here has 6. Those are each measures of alpha diversity. And then we could look at beta diversity, and that is basically a measure of how many species among these two sites, A and B, are unique to those two sites. And it turns out there are only 2 species that are unique to these 2 sites. That is, this purple flower here only occurs in site A, and this golden squirrel here only occurs in site B. And so the other species that these 2 areas have are all shared in common. And so the measure of beta diversity, or among site diversity, here is simply just 2. And then we could look at, amongst all 3 sites, how many species are unique. 1, 2, 3, 4, 5, 6, 7 species in total that are observed across all 3 of these sites. And so the gamma diversity gives us a measure of the total species richness across this entire landscape, or the total number of species that could be observed across this entire landscape. And this is kind of interesting in the context of thinking about, well, we're not just concerned with how much diversity there is in one spot. We're interested in knowing how much diversity there is across a set of different spots or different sites. And that gives us a measure of how much species diversity does a particular landscape have. Something similar is done using Sorensen's Similarity Index. Basically, it's just a metric that assesses the amount of beta diversity by comparing only the species richnesses of 2 different sites, A and B. Without getting lost in the math, I think what you should understand about Sorensen's index is if the value is particularly high, then the sites tend to share a lot of species in common. There's not a lot of diversity. If the value is pretty low, then the sites tend to be not very similar at all. And then we could look at how various geographic patterns seem to influence species richness. And one way we can view that is in the context of how much species richness there is as a function of latitude. As I mentioned, there's a lot of ecological productivity very close to the equator, and there seems to be quite a bit less as one goes poleward. And that has to do with that also influences evolution patterns of evolutionary diversification. And so this map on the or this graph on the left illustrates basically the number of plant species in a particular area or in some particular areas, and how that changes with respect to latitude, going from the equator towards the poles. And these samples were taken in all these different locations here at different parts of the globe. And what we can see is a pretty interesting correlation that species richness, at least in this case, plant species richness, tends to be highest in equatorial regions and remains relatively high near the equator. And then as we start to move towards higher latitudes, we see that plant species richness starts to decline pretty significantly. And this isn't a terribly surprising result because we know that there's a lot more ecological productivity in these sort of lower latitude regions, which probably allows for opportunities where plant lineages could diversify more so than they would in environments where productivity is limited by the availability of sunlight. We can also look at how species richness is a function of area, and this turns out to be a pretty conserved theme across different ecosystems. So if we were to start sampling different numbers of transects across an area and measuring the species richness of those different transects, we'd find that we would see so many species in, say, 100 meters of sampling. And if we ended up sampling 200 meters, we'd probably find more species. But once we start sampling 300 or 400 meters of area, we'd find that the amount of species we'd start to encounter would level off at some point to the well, really to the point where after sampling about four or 500 meters of space, we would have found all of the species that are there. At least in the context of plants, is what's shown here in this example. And this is real data taken from sampling plant communities in California, where, yeah, with only 100 meters of sampling, there were about 25 species that could be found. Whereas after sampling about 400 meters of area, about 70 species were found. But we can see that after sampling more than about 400 meters, not too many more species were found, which reflects that these surveys are sufficient to really detect the total amount of species richness that occurs in this particular kind of environment that was sampled. About 450 meters of sampling would get us at finding about all 70 to 75 species, at least plant species, that occur in these particular areas. So anyway, these are just different ways of trying to measure biodiversity and assess patterns in the distribution of biodiversity. Again, we kind of want to try to get at an understanding of what biodiversity is, and there are only so many practical ways that we can do that. Sequencing DNA to figure out genetic diversity is great, but it's expensive and kind of complicated. Whereas going out into the field and making samples of species richness and diversity is somewhat more easy, but not as somewhat easier, but not as easy as you might think. But it's probably an easier application of empirical studies. And then we could ask questions about how human population growth might affect patterns of biodiversity. And this turns out to have some pretty important consequences in the context of conservation. What do we know about human population growth? Well, human population growth is something that's been increasing since as long as there's been data on it, and it's really being driven by resource consumption. So or it's driving resource consumption, I should say. So as there are more and more people, more and more resources are needed to sustain human populations, and that inherently leads to habitat destruction because often we need certain kinds of environments in order to either grow food or extract resources from the environment. And often the combination of resource consumption and habitat destruction can drive extinction. And there are some classic examples of this with these species shown on the bottom right that have all existed during or coexisted with humans. And during the time of written history, these things were known to exist and then seen going extinct, and particularly in the context of human activities driving their extinction, whether it was hunting these things directly or simply just destroying suitable habitat for these species, they have all really been driven to extinction by different human activities. And so this is an alarming problem that conservation biologists are very focused on. How can we strike a balance between the things that are needed from the environment for human populations to be sustained, and then at the same time not completely destroy all of the existing biodiversity on our planet? And as it turns out, balancing those two things is very difficult. And as we sort of saw earlier, there's a delicate balance between appeasing the interests of different stakeholders. Conservation biologists are really interested in conserving species diversity, whereas natural resource managers or politicians might be more interested in figuring out how we can conserve resources that we might want to consume, and that inherently could lead to the destruction of ecosystems and the loss of species. And this is an ongoing problem, and there has not really been much of a great solution to this problem other than different parties involved trying to reach a compromise. And this problem is only projected to probably become worse as human population growth increases into the future. And this gets us thinking about, well, if we're driving extinction, at what rate can natural evolutionary processes restore species diversity? But we also know that evolutionary processes don't necessarily happen that fast. And so the rate of extinction often can outpace the rate of evolution. And that's particularly an issue within the modern context of conservation biology. If we are seeing species such as the ones shown on this previous slide, such as the dodo or the Tasmanian wolf, for example, if we can see these kinds of numbers of species or populations declining relatively rapidly, we can ask questions about, well, how long might it take for evolution to lead to the process of speciation, allowing that diversity to be replaced? The answer is it's probably going to take a really long time. Speciation doesn't usually happen super fast. It is a product of the generation time of different biological lineages. So organisms with a short generation time could potentially evolve more quickly than those with a longer generation time. But even then, in order for evolution to happen, some really important ingredients have to be in place. There has to be enough genetic diversity within the populations, and there has to be enough variation in sort of environmental pressures driving the process of speciation, driving the process of evolutionary change. If those things don't exist, evolution can't really happen. And again, this goes back to why considering population level genetic diversity is so important in the context of trying to understand what biodiversity is and trying to understand what we can do to conserve biodiversity. Genetic diversity is imperative in the context of allowing species to evolve in the context of environmental change. So sometimes the most practical efforts in conservation are really at the level of species. So simply trying to conserve a single species and save it from extinction, for example. And these kinds of efforts are usually focused on some kind of a charismatic taxon. For example, things like the California condor, which is probably one of the most famous conservation success stories. And so if you don't know about these birds, they're pretty interesting creatures. They're very closely related to vultures. If you look at them here, they have a morphology that's very similar to vultures, but they have an incredibly large wingspan. Some condors have a wingspan that reaches as long as 9 feet. That's pretty exceptional. These are huge birds. And these birds have an ecological niche of being scavengers. And so they've done pretty well to make a living off of sort of scavenging the remains of prey that were killed by large predators, for example. But as it turns out, the population of California condors significantly declined during the 20th century. In fact, by the year 1982, there were only 22 California condors remaining in the wild, which is an alarmingly small number. Recall, small populations are very susceptible to the effects of things like genetic drift. And so folks realized that this was not a good situation for the condors. And we can see, in fact, here this map that shows what the range of the California condor was during the 19th century. This is about 200 years ago, and what that range looked like, shown in the little red area here, by the 1940s. And so these condors experienced significant range contraction as human populations started to settle Western North America and really overtake the area of suitable habitat for these condors, in addition to some other human activities that indirectly had negative effects on these condors, such as lead poisoning. So these condors, as scavengers, would often eat things that were left behind by human hunting activities, where people had shot game animals with lead bullets. And so these condors would inadvertently consume those lead bullets, making them sick and often leading to fatalities. And as California condor population sizes started to decrease and decrease and decrease, their low population size led to decreases in their genetic diversity, which started to make them a lot more susceptible to diseases. And so these things kind of worked hand in hand to sort of drive California condors towards extinction. And so again, by the early 1980s, folks realized how bad of a situation this was. And so the last remaining 22 condors were taken into captivity and raised in captivity. I think it was at the San Diego Zoo. And they stayed in captivity for a while until their populations in captivity were doing okay, and they were breeding in captivity. And so folks thought, okay, we're seeing population sizes increase in these condors in captivity. Let's go ahead and see what happens if we release some of them back into the wild. And lo and behold, these things were released into some certain conservation reserves or conservation preserves, and they were able to begin breeding successfully in the wild. And today we see that, in fact, the number of the number of wild California condors has exceeded the number of those that are in captivity. So this is a success story of how conservation efforts can work if the problem is identified in time before population sizes become too small to do anything about it, and if captive rearing programs are successful in allowing lineages of threatened species to continue to reproduce, and if areas of suitable habitat can be recognized and protected as areas where we can release these small threatened populations back into the wild. So this really represents an idealized situation where conservation efforts were successful. As I mentioned, again, small populations are especially susceptible to the effects of genetic drift. And basically, if you don't remember, genetic drift is a random change in gene frequencies that happens in populations. And it happens in populations all the time. It just turns out that the effects of genetic drift are a lot more noticeable when there are less reproductive events in populations, which basically means populations that have fewer individuals are going to be more affected by genetic drift than populations that have lots of reproductive events. And so when there is low genetic diversity, not only does it make it harder for species to adapt to environmental change, but it leads to inbreeding. And inbreeding is basically something that happens when very closely related individuals start mating together. The graph here on the top right shows an example of the effects of inbreeding on basically the fecundity or fertility of offspring involved. So this is data taken from this small wildflower in the genus Silene. And what we can see are the germination successes of plants taken from different kinds of crosses. And these crosses involve crosses where the parents were very distantly related, so the level of inbreeding is quite low, compared to crosses of parents that were very closely related, where the level of inbreeding is quite high. And what we see pretty clearly looking at this graph is that germination success goes down quite significantly as the level of inbreeding starts to go up. And this is a classic example of why it is that the offspring of closely related individuals have a lot less fitness or a lot less success than the offspring of parents who are distantly related. And so why is this happening? A lot of this has to do with the fact that when closely related individuals mate with each other, it increases the probability of harmful combinations of genes linking up during fertilization. And this is not a great thing. This makes populations have less reproductive success. It makes them more susceptible to environmental change because basically everybody in an inbred population is carrying this a very similar, if not the same genome, which can make them more susceptible to diseases. And so there are multiple reasons why inbreeding is not a good thing for populations. And it tends to be something that sort of inherently happens when population sizes are relatively small. Probably these California condors in the early 1980s were beginning to suffer from the effects of what we call inbreeding depression. And they were fortunately able to overcome that. However, typically what happens when population sizes crash as a result of, say, human activity is that genetic variation crashes with population size, which isn't surprising. Fewer individuals, lower probability of having a lot of genetic diversity in populations or a lot of genetic variation in populations. And even though population sizes can rebound after a population crash, genetic variation usually takes a long, long time to become restored. And you might wonder, why is that? Well, first of all, you have to think about what are the sources of genetic variation. Well, the only source of genetic variation is genetic mutation. And we know that most mutations either have negative effects or neutral effects, and the chances of beneficial mutations arising in populations aren't great. And so it takes a long, long time for beneficial mutations to arise and restore good genetic variation within populations. Another way we could see genetic variation restored in populations is through gene flow. We'll talk about in a moment. But effectively, if there's not a source of genetic variation from which gene flow can enter small populations, then all those populations have is are the chance mutations that might arise that are beneficial to those populations. And that's very much the case we see with these condors. There were no other populations out there to contribute genes or to promote genetic admixture. And so even though California condor numbers have rebounded following their population crash, we could ask important questions about, well, how much genetic variation is there in the current populations of California condors? And the answer might be that there is not very much. And I had mentioned this thing, gene flow. So what is gene flow and why does it matter in the context of conservation? Well, gene flow simply is just the movement of genes in and out of populations, or it's really the product of migration and subsequent mating. So if we have, for example, two populations, as shown in this cartoon, we could talk about gene flow being the situation where, for example, individuals from population 1 migrate over to population 2 and mate with individuals from population 2 and start to mix up the gene pools of these two separate populations. What are the gene pools? Gene pools really just represent the sum of all of the different genes in a particular population. And ideally, if populations have a diverse gene pool, they have a lot of different genes, and that can allow them to, again, deal with things like environmental change or diseases more readily than populations that have relatively restricted gene pools. And so often gene flow is associated with overcoming barriers to migration. So this cartoon illustrates how a mountain range, for example, might be a barrier to gene flow between population 1 and population 2, which in this case are represented by these western and eastern deer populations. And so frequently in the context of conservation, we think about how is it that we can allow populations that might be threatened to overcome barriers to gene flow. In this case we see that these deer are able to squeeze through a mountain pass, and they're able to overcome this barrier to gene flow, and that allows the gene pool of population 1 to become somewhat more diverse, and likewise the gene pool of population 2, because these two populations are basically sharing genes with each other. And this promotes what we call genetic admixture, which is a fancy way of saying if two or more populations are exchanging genes with each other, then there's a higher frequency of genetic variation amongst populations than there is without genetic admixture. So gene flow is really, really important because without lots of good mutations arising all the time, this is the only way that populations can accumulate more genetic diversity. And again, we know that genetic diversity is important for populations because it allows them to deal with changes in the environment. It allows them to deal with being able to resist certain diseases. Gene flow is inherently a consequence of something called dispersal, and that's the process by which individuals from one population are able to disperse to either new areas or disperse to share genes with members of other populations. And this can happen in some different ways. We can talk about density-dependent dispersal, which is shown with these grasshoppers here, and effectively density-dependent dispersal happens when there are so many individuals that occur within a particular area that population density becomes really high. And when population density is really high, there aren't really enough resources to sustain all those individuals. And so this prompts a subset of that population to disperse away to a new environment where resources are abundant. And this prevents members of the population from dying off and allows all these individuals to survive by dispersing to new areas. Other forms of dispersal that we might talk about in the context of plants are things like anomachary. This is wind dispersal. So, for example, plant seeds or plant fruits rather often have adaptations such as things like wings or tufted hairs, which allow them to travel in the wind. So dandelion fruits are a classic example of these wind dispersed fruits that have these little tufted hairs that will allow the fruits to be picked up in the wind and allow those fruits to become established in new areas where there might be other dandelion populations, and these dandelion populations could potentially share genes. There's also something similar called ectozoochory, which is a situation where perhaps the fruits of plants stick to vectors like this dog here, or like animal vectors such as mammals that have hair, where these fruits are sort of adapted to stick to the hair pretty well. This is a cocklebur fruit, by the way, here. And you can see that this cocklebur fruit has these little burrs on it, and those burrs are really effective at clinging to hair. And this dog, who has traveled through a whole field of cockleburs, is going to take these cockleburs somewhere else, probably disperse those fruits off, and the fruits will eventually release the seeds. And now a new population can either become established, or a small subset of another population can disperse to a new environment where they can share genes with members of other populations. And then a similar process where animal vectors can move things around is known as endozoochory. That's like digestive dispersal. So, for example, this bird might eat a praying mantis and it'll kill the praying mantis and digest it. But let's imagine that that praying mantis is a female and she's bearing fertilized eggs. Well, as it turns out, those eggs are resistant to digestion. And so even though the mother is killed by being eaten, her eggs survive. And so this bird travels off somewhere kind of randomly and basically poops out the remains of this meal. And some of those remains include the eggs of that female. And those eggs, which were able to survive digestion, then hatch and give rise to the offspring of that praying mantis. And now a whole new group of praying mantises have been dispersed into a new environment where they could potentially share genes with other praying mantises. So all these kinds of processes are processes that promote gene flow between populations. Really, they're promoting dispersal, and by promoting dispersal, they are increasing the probability that different populations are going to share genes. And this really is a good thing for these populations because it's helping to increase genetic diversity within their respective gene pools. And as I sort of mentioned, barriers to gene flow can prevent genetic admixture between populations, and that's not really a super great thing. How does this happen? Well, often this is driven by a process called habitat fragmentation. Now, this could happen naturally, that is, in the absence of human influence, but also it's more frequently something that's thought of as being driven by human activities. And so, what's habitat fragmentation? It's basically a reduction in the area of suitable habitat that exists for populations. And so, we can sort of look at this cartoon on the left, and the cartoon illustrates what might be an area of good continuous habitat for a species. compared to a similar area that's been fragmented or broken up into a bunch of substituent little parts. And so what's going on here is that we might have an area with really, really good continuous interior habitat. And typically that area of interior habitat is surrounded by something we call edge habitat. And usually interior habitat is of higher quality than edge habitat. And when an area of continuous interior habitat is broken up into subsequent smaller pieces, it causes the area of edge to increase. That is, the amount of interior habitat goes down and the amount of edge habitat goes up, or the amount of good quality habitat goes down and the amount of poor quality habitat goes up. If we want to think about this in terms of geometry, we can look at this image on the right here. And let's imagine we have an area of interior habitat that's represented by about 64 hectares, which is just a measure of area, kind of like acres. I think a hectare is 2.5 acres. Anyway, we have 64 hectares of good, suitable habitat represented in this square. If we took that same square and broke it into 4 smaller squares that were each only of 8.7 hectares each, we can see how inherently the amount of edge is going up. We went from having 64 hectares of good, suitable interior habitat to now only having about 35 hectares of good interior habitat. And in the process, there's a lot more edge. And as this example shows, there are human activities that have driven this fragmentation. The building of a railroad, for example, or the building of a highway, for example, that has taken this good interior habitat and broken it into 4 smaller patches of interior habitat. And there are all kinds of human activities that can cause this. Things like urbanization, obviously, is associated with road building. Things like agricultural activities are commonly associated with driving habitat fragmentation. Again, building things like pipelines or canals, or things like energy transmission corridors, or things like the high-speed rail that's being constructed in Southern California, or in California right now, I should say. These are all things that can contribute to habitat fragmentation, which really leads to a loss of good suitable habitat for populations and an increase in the amount of edge habitat. And a really important question that arises in the context of habitat fragmentation is whether or not populations can overcome these barriers to gene flow. An area of good continuous habitat is now broken up by things like roads or highways or railways, for example. Can populations cross those barriers or not? And that depends on the kinds of organisms. Things that travel across the ground might have a hard time crossing those kinds of barriers. Things that can fly might not have such a hard time, or things that can disperse in the wind might not have such a hard time. So it really depends on the kinds of species that we're talking about, but not all species are going to be able to overcome barriers to gene flow so equally. And so habitat fragmentation is really an important cause of concern in the context of conservation. And typically what happens after there is habitat fragmentation is that we can think of what once was a wide-ranging population getting broken up into a bunch of smaller populations. And there's a term we use for that. It's called a metapopulation. And a metapopulation is sort of this fancy term we use to describe a population of populations. So a population that might have been one continuous population previously, as a result of habitat fragmentation, is broken up into a subsequent bunch of smaller populations that we would call a wide-ranging metapopulation. And metapopulations are typically described in the context of the size of the patches that they occupy and the isolation of those patches. And so we can sort of view metapopulations as being those that occupy relatively large patches with relatively high amounts of connectivity between them. And that's kind of the best case scenario for managing a metapopulation. Whereas a sort of worst case scenario are metapopulations that have relatively small sizes that are really isolated from one another. And what we can see is that when that happens, the probability of genetic admixture between different populations is going down because of their isolation, and because by having smaller patches, they are probably going to harbor smaller population sizes, and smaller populations, since they're more susceptible to the effects of genetic drift, are probably going to accumulate lower levels of genetic diversity. And so often when managing metapopulations, conservation biologists are focused on keeping these metapopulation patches relatively large and relatively more or less connected. Oftentimes, we can think about metapopulations in the context of what we call source and sink populations. And so source populations are typically what we might call sources of genetic variation, whereas sinks are often areas where individuals might disperse to, but that's kind of the end of the line. It's sort of like a one-way street. So for example, if we look at this particular metapopulation here, the big blue circle represents a source of genetic variation, and the arrows represent individuals dispersing from that source into smaller patches, and we only see those arrows moving in one direction. Genetic diversity is not coming from the small patches back to the large patch. Genetic diversity is only really going from the large patch to the smaller patches. So this large patch is a source, and these smaller patches are sinks. So this is a hypothetical framework, but it's very much the way that the management of fragmented populations is viewed. And we can look at a real life example of this occurring in a population of ungulates here in California. So the desert bighorn sheep are a well-known example of a species that is particularly threatened and composed of a pretty wide-ranging metapopulation. Sheep species in North America generally had a lot larger populations in the past, but they've been threatened, particularly by things like diseases. As domestic sheep from Europe were introduced to North America, a lot of native North American sheep couldn't resist those kinds of diseases. And also, we see that nowadays human activities are also causing or presenting barriers to gene flow amongst sheep populations, causing them to have smaller population sizes, which are susceptible to losses of genetic diversity. So this isn't a very good situation, and the desert bighorn sheep is a particularly sensitive example of this phenomenon. This map here shows the range of desert bighorn sheep metapopulations, and so each of these different colors represent different populations, and the numbers represent their population sizes. And if you don't know, each of those colored blob-looking areas, which represent populations of bighorn sheep, also represent mountain ranges where those bighorn sheep have suitable habitat. And these mountain ranges are typically pretty rugged places. And those are places where these bighorn sheep do pretty well to sort of execute their normal life history strategies. But as it turns out, if you look at these numbers, you'd see that a lot of these populations aren't particularly large. And if you look at the little arrows, you can see where corridors of gene flow exist between these metapopulations, between different metapopulations here. But there are certainly areas where there seems to be no gene flow between these populations whatsoever. And so the probability of gene flow between these different populations really does seem to depend upon their proximity, how close are they to one another, and whether or not there are significant barriers to dispersal between these populations. So things like these highways, each of these black lines here represent major highways that humans have built. And often these sheep will not cross these highways for obvious reasons. Would you cross one of these highways? Probably not on foot anyway. And so these sheep will avoid human-made barriers to gene flow, and that restricts gene flow between their populations and contributes to subsequent losses in their genetic diversity. Not a great thing for these sheep. How do we know that? Well, there is actually data on the survivorship of these small populations. And as it turns out, these desert bighorn sheep have been very susceptible to what we call extirpation or local extinction. And this graph here kind of illustrates how that situation has played out during the 20th century, where we can look at 5 different desert bighorn sheep populations whose survivorship was tracked over the course of 50 years and see how their population sizes were directly related to their survivorship. And this line on the top represents populations whose sizes were greater than 100. This blue line represents populations whose sizes were greater than 50. This orange line represents populations whose sizes were between about 30 and 50. The green line represents populations whose sizes were between about 16 and 30. And the black line on the bottom represents populations with a size of 15 or less. What do we see looking at this graph? Well, the percent of populations that were able to persist was certainly greatest in the desert bighorn populations that had the largest size. Those with a size of 100 or 50 or more, I should say those with a size of 100 or more, or those with a size between 50 and 100, were able to persist over this time period within which their survivorship was studied. But the populations that had a size of 50 or less, none of those populations were able to survive. They all went locally extinct. And that's probably because they were suffering from inbreeding depression, suffering from the effects of loss of genetic diversity due to genetic drift, increasing the odds of harmful combinations of genes pairing up. These populations were really highly inbred. And so they just weren't able to survive this particular effect of loss of habitat area causing their population sizes to become so small. So this is a real example of the effects that habitat fragmentation can have on small threatened populations that not only causes them to lose genetic diversity and increases inbreeding, but it's also decreasing the probability of gene flow between them. We can likewise look at an example that illustrates why it is that gene flow can really be important to the conservation of genetic diversity for populations. And as it turns out, really just a little gene flow can be important. This example shows data taken from the mountain lion populations that occur here in the Santa Monica Mountains and Simi Hills, not too far from the San Fernando Valley. So California State University Northridge is out here somewhere. And here's Malibu over here, and here's Ventura over here. And all of these different colored sections on this map represent the geographic range of different mountain lions that were tracked by biologists. I think they used the radio collar method, so they had GPS coordinates of all the different areas where these mountain lions were traveling. And if you look at that map, what you realize is that a lot of these mountain lions were range restricted. They didn't seem to travel very far beyond a certain area of suitable habitat. And in particular, we can see that individual mountain lions that occur north of the 101 freeway seem to exclusively occur north of the 101 freeway, whereas individual mountain lions that occur south of the 101 freeway seem to exclusively occur south of the 101 freeway. And so what is the 101 freeway? Well, it's a barrier to gene flow, preventing admixture between mountain lion populations north of that barrier and those south of that barrier. And this isn't a great thing for these mountain lion populations. As it turns out, a single individual mountain lion who was assigned the name P12 was able to cross the 101 freeway. And after P12 crossed the 101 freeway, P12 was able to successfully mate with a number of different females, or a couple different females, south of the freeway. And what we see on the left is basically a genetic structure analysis of these different populations of mountain lions. And effectively, this is kind of complicated, but what these different bars each represent are individual mountain lions. So for example, this is P1, this is P2, this is P5, this is P6, et cetera, et cetera. And the colors represent basically the percentage of genetic cluster assignment. All you really need to know is that individuals who share the same colors tend to share a lot of the same genes in common. So for example, these individuals all shown in blue are very genetically similar, whereas these individuals who have some green proportions within these bars are a little bit more genetically similar to each other. And in contrast, these individuals who have a high proportion of red shown in their bars are more genetically similar to each other. And so we can see kind of 3 distinctive kinds of genetic signatures in the populations of mountain lions that were surveyed. And these populations on left are all the populations in the Santa Monica Mountains. And these populations here show those that occur in the Simi Hills, sort of north of the 101 freeway. And these populations over here, mostly represented by red, are those that occur in the Verdugo Hills and the San Gabriel Mountains, which, if you know, is a little bit to the east of our map here. It's not shown on our map, but it's not terribly far from where the 210 freeway is, or where the 210 and the 5 freeways sort of are to the east of the San Fernando Valley. And so anyway, it looks like the mountain lions of the Santa Monica Mountains are all mostly genetically similar to each other when compared to mountain lions from the Simi Hills that are mostly genetically similar to one another, and likewise when compared to mountain lions of the Verdugo Hills and San Gabriel Mountains that are mostly genetically similar to one another. And why are they mostly genetically similar to one another? Because they are dealing with these barriers to gene flow. These individuals from these populations don't very frequently breed with individuals from these populations, who likewise don't very frequently breed with individuals from these populations. But as I mentioned, P12 was able to cross from the Simi Hills into the Santa Monica Mountains. And we can actually see P12 represented right here by this green bar. So the genetic makeup of P12 was very characteristic of the population from the Simi Hills. And what we see shown by these individuals here are the genetic makeup of all of P12's offspring. So P12 had 1, 2, 3, 4, 5, 6, 7, 8 different offspring. And what we can basically see looking at this is that P12 was able to take some of his genes and bring them into the population of mountain lions in the Santa Monica Mountains. And so all of P12's offspring have a lot more genetic variation in their genome than individuals that were the product of matings from only members of that population in the Santa Monica Mountains. And so by just one individual P12 crossing this barrier to gene flow and mating with a few different individuals, we were able to see an increase in genetic diversity in this mountain lion population. And that's a very good thing for this mountain lion population, because it's allowing them to effectively deal with these barriers to gene flow by allowing their genetic diversity to be increased. And so if we apply our knowledge of the importance of gene flow for populations and the importance of genetic diversity within populations, particularly in the context of threatened populations, small populations, we can start to think about how it is that protected areas get designed. That is, areas that are have the goal of conserving suitable habitat for populations that might be threatened. And so if we take this thinking about metapopulations and how they are particularly sensitive to the effects of habitat fragmentation because of barriers to gene flow and because of the effects of genetic drift on small populations, we can start to think about the most effective strategies for designing protected areas, or areas that we might use to preserve or conserve species. And so as it turns out, protected areas function a lot like islands. And we can apply something known as the island theory of biogeography to thinking about how we can best manage biodiversity within protected areas. And so in short, the island theory of biogeography kind of is framed in the context of the size of suitable areas and the proximity of suitable areas to source populations. And the way this basically works is how the number of species that can potentially migrate is an artifact of the size of patches and the proximity of these areas to one another. This is kind of viewed in this cartoon here, where we have some mainland environment. We might call that the source population. And then we can look at these different islands off the shore of this mainland that kind of represent potential areas to which these different populations can disperse. And so the idea is that we can have large islands that are relatively close to the mainland, or small islands that are close to the mainland, or small islands that are far from the mainland, or large islands that are far from the mainland. And this is predicted to influence how many species these different islands can harbor. And so this graph basically shows how island A, which is kind of small and far from the mainland, would be expected to have the lowest species richness, whereas island B, which is also kind of small but a lot closer to the mainland, would have a little bit higher species richness, whereas island C, which is kind of large but still kind of far from the mainland, would still have a little bit higher species richness. And then island D, which is the largest, which is a large island and very close to the mainland, would be expected to have the highest species richness. And so if we took this model and put it into the context of, instead of thinking about mainlands and islands, we instead think about source habitats and so-called sink habitats, we can start to predict how these different areas might be able to harbor different levels of biodiversity depending on their size and proximity to a particular source population. And this leads to something that is classically known in conservation biology as the SLOS debate, or the single large or several small debate. In other words, is it better to have a single large protected area, or is it better to have several small reserves? And there are different caveats to either situation. We might think that having a single large protected area is better because it's an area of good quality continuous habitat. Whereas we might instead think that having several small protected areas is better because maybe those different environments can harbor different kinds of biodiversity. And so we might have higher gamma diversity or higher landscape level biodiversity conserved with several small reserves considered when compared to a single large reserve. Or not. There's only really one way to find out what would be better, and that would be to actually put these theories into practice. And so this is very much what the science of conservation is focused on, is trying to figure out what is the best and most effective way to design protected areas. And so, as it turns out, an idealized design is certainly not always achievable for various reasons. Suitable habitat can only be secured and protected in certain areas, or there has to be a balance between appeasing different stakeholders. Again, this is kind of a common theme in conservation. Biologists might want one thing, resource managers might want another, and this kind of dictates which areas are going to be protected and which areas aren't. And so oftentimes this leads to mixed use zoning. And mixed use zoning is often necessary to keep all of the parties involved in managing resources appeased. And so, for example, there might be various goals in mind, such as maintaining tourist industries or recreation industries. Certainly, there are scientific goals, such as monitoring natural populations, doing research on the reproductive success of natural populations. And some of these goals also might involve things like education, using these areas for perhaps teaching biology students or students of other natural sciences things about conservation, or training young practitioners in various agencies about the things that are done in order to maintain conservation in these areas. So, for example, a protected area might be designed like this, shown on the left, where there are transition zones between highly urbanized areas and protected areas where tourism and monitoring and education are all sort of happening in equal proportions. But then as we move to the interior of this protected area, we get to a buffer zone where there's still limited amounts of tourism and recreation and increasing amounts of monitoring and research, and maybe increasing amounts of education. And then at the true interior core of this protected area, where the largest continuous area of good quality suitable habitat exists, that area is really only designated as an area for monitoring and research. And so if you've ever been out into certain protected areas like national parks, for example, you might have noticed there are areas that are open to tourism, and there are areas that are sort of off limits because these are areas that are particularly sensitive, where perhaps small populations might be given a limited area of suitable habitat that is restricted from any human activities other than simply scientific research. So that's one kind of goal. Despite, however, these particular protected areas might get designed kind of inherently because of the way the landscape is oriented, really the most important goal from the context of biologists is to maintain connectivity between populations. And so whether or not we are going to deal with having a single large preserve or several small preserves, really one of the most important goals is making sure that there is habitat connectivity between these areas so that different small populations or metapopulations can exchange genes with one another and maintain relatively high levels of genetic diversity. And so we can kind of look at comparing and contrasting some different artifacts or different features of protected area designs and what might be worse and what might be better for different reasons, such as the size of the reserve. Obviously, a larger reserve is going to be better than a smaller reserve because there's simply just more area of suitable habitat. Obviously, an intact reserve is going to be better than a fragmented reserve because there will be less edge and a larger area of continuous suitable habitat. Probably having several reserves is better than having a single reserve, irrespective of their sizes. More area is generally, or more different areas, I should say, is going to probably be more effective than having less areas. Having areas of different sizes could turn out to be important for different reasons. But this could have to do with the diversity of these different areas and the diversity of kinds of organisms that could survive in areas of different sizes. Again, connectivity is key. Having isolated reserves is not nearly as effective as having reserves that are somehow or other able to promote the connectivity or the amount of gene flow that's going on between them. Having diverse ecosystems is a lot better than having relatively uniform reserves or uniform ecosystems where we see sort of the same habitat repeat itself over and over again. It would be better to have a reserve that encompasses lots of different kinds of habitats, which is part of the reason why having reserves of diverse sizes could be important. Having fully protected ecosystems is key, whereas partially protected ecosystems, if they're interfaced with urban areas or areas where there's agricultural or industrial activities going on, it presents the opportunity for pollution or toxins to enter a protected ecosystem, for example. So trying to conserve an entire ecosystem that is not at all influenced by negative human activities is generally key. Having relatively round reserves tends to be important, or basically reserves of a nice shape rather than a sort of thin shape is better because it again reduces edge effects. So a really linear reserve is going to have a lot of edge, a lot of area of not very suitable habitat, whereas a very round reserve has less edge. And that's simply an artifact of the geometry of the protected areas designed. And coordinated management is pretty important. So if we had a bunch of different reserves, but they're all sort of managed individually, and none of those agencies are communicating in terms of their management policies, that's not nearly as effective as coordinating the management of these different protected areas. Even if they are managed by different agencies, those agencies should be communicating and coordinating their efforts. And generally, trying to exclude humans from protected areas is typically a good thing. Some folks are good and have good intentions when they go out into wild areas, but some folks don't. Some folks appreciate the importance of conservation, but other folks don't. And so integrating people into preserves is important for educating them, but we have to be careful about the extent to which humans are able to interface with protected areas because human activities can, of course, threaten protected populations, especially if they're very small and sensitive. So there's a lot of complicated things going on when the design of protected areas are considered, but we do need to think about all these potential factors that are at play when designing protected areas. So that's basically it for our treatment of conservation biology. This is very introductory, but I tried to touch on the central themes in conservation, again, which are things like thinking about what is nature, what is conservation really about, how is that different than preservation, how it is that this is framed as a crisis discipline, really at this interdisciplinary junction between different kinds of management systems, both scientific and non-scientific, how conservation biology is really oriented around trying to protect biodiversity at all of its different levels and how it is that we can measure biodiversity. Also, how it is that we can deal with trying to prevent species extinction if possible. And of course, again, how it is that conserving genetic diversity is really one of the ultimate and most important goals in conservation. And how is that maintained? Well, it's maintained by keeping population sizes relatively large, promoting gene flow between them, and keeping those populations basically genetically healthy. And then how it is that human activities can influence the availability of suitable habitats for populations. So these are all sort of the overarching themes that are viewed in the context of conservation biology and are very, very important themes that are viewed in the context of managing natural populations. So that's it for this video. I'll see you next time.